Okay, so this is quite an important lecture. It tells you basically how different genes in your body are regulated. Obviously, I think we all know that every cell in the body, every cell in the human body has the same genome, right? We all have the same DNA, and we have 20 to 25,000 genes. But from that 20 to 25,000 genes, some cells become muscle cells, some cells become immune cells, some cells become adipose tissue or fat cells, some cells become part of the bone, some cells become nerves, and some cells become blood cells, like red blood cells carrying oxygen. From that, but they all have the same gene. From that, you should pretty much, you can guess that these cells express these genes differently. So for example, an immune cell requires voltage-gated sodium channels, right? So to activate action potentials. So it will activate specific genes which allow it to carry out its function. That's a better way of putting it. The liver is responsible for detoxification. So the liver cells or hepatocytes, they synthesize genes which are responsible for detoxification. No, we've just started. All right. So this is the point here that all your cells in the body have the same DNA, they have the same genome, but they regulate and express these genes differently to perform their function. And this idea is called differential gene expression. Okay. So each cell in the body has a different expression of gene. This is called differential gene expression. I will kind of center the mechanisms we discuss in this presentation around cancer development. So let me just kind of explain these things to you basically. And you don't need to kind of go into depth with this, but you should know that normal cells regarding cancer have two types of genes. One are called tumor suppressor genes, and one are called oncogenes. All right. So one set of genes are called tumor suppressors, and one set are called oncogenes. These tumor suppressors in a normal cell are upregulated or expressed. And these oncogenes, which are cancer promoting genes, they're normally silenced. In a cancer cell, so this is normal, right? And in a cancer cell, what will happen is that these tumor suppressor genes will go down or become silent. They won't be expressed. And these oncogenes or cancer promoting genes will go up. And so you should know the mechanisms behind how this happens, because many cancer drugs out there that you'll study later on, they work through basically normalizing this gene expression pattern. They allow the cancerous gene expression to go back to a normal cell. And this is the way they function. All right. So keep in mind these two sets of genes in cancer cell, and we'll kind of talk about them later on. So I think you've seen this slide beforehand. This is the central dogma of molecular biology. This is DNA, your genetic code. You can replicate it by an enzyme called DNA polymerase. This DNA can also get transcribed into messenger RNA by an enzyme called RNA polymerase from mRNA. mRNA goes from the nucleus to the cytoplasm, where it's bound to ribosomes and gets translated. All right. So these two steps, transcription and translation, they are what define gene expression. Okay. So if someone tells you or asks you what gene expression is, the simple answer is transcription plus translation. The reason why you should also get this definition straight in your head is because it also explains how it's regulated. There's two steps to gene expression, right? Transcription and translation. There's also two ways you can regulate gene expression. One is transcriptional regulation, one is translational regulation. Transcriptional regulation determines how much of that DNA gets made into mRNA. So how much mRNA you have is determined by transcriptional regulation. Translational regulation determines whether that mRNA gets successfully translated into proteins. All right. So gene expression is transcription and translation. And these are how you also regulate gene expression, transcriptionally or at the translational level. So this is a general schematic of DNA gene expression. You have DNA here. It gets transcribed into mRNA. First, it gets made into pre-mRNA and then undergoes processes like capping and polyadenylation and splicing to form mature mRNA. This mRNA goes into the cytoplasm to get translated into proteins, all right? And you can regulate this at many levels. You can regulate it at the transcription level, which we discussed, by determining how successful is RNA polymerase at transcribing this DNA. You can also regulate it at the translational level by determining how much of this mRNA gets translated into proteins, all right? And so these are the two things we'll talk about today. So let's discuss transcriptional regulation first, all right? There's two types of transcriptional regulation. And some of you should kind of write this down, because if you write this down, it'll help you kind of keep up with the lecture. So you have gene expression. Gene expression can be regulated transcriptionally or translationally. Transcriptionally, there's two types. One is called epigenetics, and one is through DNA binding proteins, which are transcription factors. So there's two types of transcriptional regulation. Then there's two types, further subtypes of epigenetics regulation. One is acetylation slash deacetylation of histones, and one is methylation or demethylation of promoters. Okay, and the DNA binding proteins, they refer to transcription factors. And transcription factors are two types, general or specific. This is kind of a general overview slide. We'll go into now each of these mechanisms in detail. But as a general point, there's two types of transcriptional regulation. One is epigenetics, one is transcription factors. There's two types of epigenetic regulations you should know. One is histone modifications, acetylation, deacetylation. One is methylation or demethylation of promoters. The transcription factors are also of two types, general or specific. Okay? So this is what I just told you in these last few slides. The central dogma of molecular biology is gene expression, i.e. transcription plus translation. All these cells have the same genome but express it differently, and this is essential for cell function and cell identity. And this is called differential gene expression. By this definition of gene expression, you can also tell how gene expression is regulated. It can be either transcriptional regulation or translational. Transcriptional regulation includes epigenetics, histone acetylation, deacetylation, and transcription factors, general or specific. So let's talk about histone modifications first. So you have histones here. Histones are proteins which bind to DNA. They're found in groups of 8. Those 8 histones are made up of 2 copies of 4. So there's 4 histones. You have 2 copies of each, which give you 8. Around these 8 histones, DNA is bound to form a nucleosome, all right? And you have 4 types of histones, H2A, H2B, H3, and H4. You have 2 copies of each, so you have 8 in total. DNA is very, very tightly bound around these histones to form a nucleosome, all right? So now, how can you regulate histones through epigenetics? Let me just show you on a screen here. Suppose you have a histone. Let's call this histone H3, all right? This histone has a tail, and this tail is very rich in lysine, the amino acid lysine. It has a lot of it. The thing about lysine is it's positively charged, okay? So let's say lysine is very, very positively charged. The thing with DNA is it's negatively charged because deoxyribonucleic acid. Acids are negatively charged. Because of this positive negative charge of histone and DNA, there's a very strong attraction between DNA and histones. So the DNA is very, very tightly bound to histones because of these electrostatic attractions, right? So let's draw the DNA here. Because of this positive negative attraction, DNA is very tightly bound to histones. Now imagine if I add a acetyl group, which is CH2, CH3. This group is nonpolar. It doesn't have a charge. So if I add it to the histone, this positive charge will decrease. If this positive charge decreases, then the attraction between DNA and histones will get weaker because the positive charge is decreasing, DNA is negatively charged. Hence, the electrostatic attractions between histones and DNA will get weaker if I add an acetyl group to a lysine. What will happen as a result is you have a histone here, you have the tail, and you have DNA which is very, very loosely bound to the histone. Because of this, RNA polymerase, which is the enzyme responsible for transcription, can very easily access these genes. Normally, these genes were very, very tight to the histone. It was very difficult for RNA polymerase to come in, bind to these promoters, and transcribe these genes. But because now you've acetylated these lysine tails, you've loosened up the DNA chromatin structure, and you've made it accessible to RNA polymerase for transcription. So acetylation will increase gene expression. Does this make sense to everyone so far? You can type in the chat. I can read the chat if you want.
Yes.
Okay, that's good. So acetylation increases gene expression. And you also know this from your basic histology because you know that there's two types of chromatin, right? You have euchromatin and heterochromatin. Heterochromatin is very, very tightly packed and it's not expressed. Euchromatin is very, very loosely organized and so is expressed. So euchromatin, which is expressed, has acetylated histones. Heterochromatin, which is not expressed and is dense, that has deacetylated histones, or their lysine tails don't have histones, and so DNA is very, very tightly bound of heterochromatin. Hence, heterochromatin is not expressed. Okay? So let's go back and read what I said here. Histone tails protrude outwards to form a nucleosome. These are the unacetylated histones. The blue is DNA, the purple are histones. You see how tightly they're wrapped around? Now you add lysine to this. This green thing that you see is acetyl added to the lysine tails. And you can see how DNA kind of loosens up. And these are acetylated histones. And acetylation of histone tails promotes loose chromatin structure, which allows transcription. And that's what I've said here. Histone tails contain positively charged lysine, resulting in strong electrostatic attractions with negatively charged DNA. Okay, someone wants me to repeat. I'll repeat this, no problem. So I'll repeat this right now. So histone tails contain positively charged lysine and DNA is negatively charged. And so there's a very strong attraction between histones and DNA. Acetylation, which is this group, this is an acetyl group. If you add this to the lysine, that will reduce the positive charge of the lysine and that will weaken these DNA histone attractions. This will loosen up the chromatin and make it easier for RNA polymerase and transcription factors to access. And so this will increase gene expression. Deacetylation has the opposite effect. It will tighten up the chromatin structure into a heterochromatin. The enzyme which causes acetylation, as tells you the name tells you what it does, is called histone acetylase. The enzyme which removes an acetyl group is called histone deacetylase or HDAC. Okay, so this is histone acetylation. What stage is this happening at? So this is before you express the gene. This is DNA, and you're determining whether it gets expressed, whether it gets transcribed or translated. Whether it gets transcribed depends on how accessible it is to RNA polymerase. If it's very, very tightly bound with unacetylated histones, RNA polymerase can't access it. If it's very, very loose, that means RNA polymerase can access it and can transcribe it. This is pre-translation. This is pre-transcription, okay? This is before the gene is expressed or touched by RNA polymerase. Transcription is from the gene to mRNA. Translation is mRNA to protein. This is before mRNA. This is before transcription, okay? I'll have a kind of separate section for questions, and so you can type in the chat, and then I'll kind of go through the question. So that's histone acetylation done. Let's talk about DNA methylation. Let me give you the same example on the white screen. So you have a gene, you have a promoter of a gene, and you have the rest of the gene. This is the 3' end of a gene. This is the 5' end of a gene on DNA. This is the promoter. RNA polymerase, this is the promoter, right? RNA polymerase has to bind to this promoter to make mRNA. mRNA. This promoter, however, is very rich in cytosine. So there's 4 bases you know, right? Adenine, guanine, cytosine, and thymine. The promoter is very, very rich in cytosine. What you can do is you can add a methyl group to cytosine here or in here, and you can add a methyl group here. This will prevent RNA polymerase from binding to this promoter. If RNA polymerase doesn't bind to the promoter, this will shut off transcription and this will silence gene expression. So DNA methylation silences gene expression or decreases gene expression. Okay, we have a question. Why is this the case? Could you perhaps unmute your mic and I can answer you? Because I don't really understand your question.
I mean, why does methylation prevent the RNA polymerase from binding to the promoter?
Because RNA polymerase is a protein which recognizes a specific base sequence. And if you change this base sequence by methylation, RNA polymerase won't be able to recognize that specific sequence. Does that make
So
sense?
it's not complementary, you mean?
Yes, yeah, exactly. So RNA polymerase requires the promoter to be in a specific conformation or in a specific structure. The methyl group will kind of interfere with RNA polymerase promoter interactions and will prevent RNA polymerase from binding to the promoter. And so it will silence gene expression.
Okay, thank you.
Okay. So we have another message. Methylation and deacetylation suppress gene expression. Yes, both of them suppress gene expression. Acetylation and demethylation will increase gene expression. Okay. Now, so this is kind of a better diagram explaining it. You have a promoter here, you have the gene here, you have the cytosine residues here. They are unmethylated. So you have transcription factors binding to these promoters, and then RNA polymerase binding to this whole complex, which causes gene transcription. When you methylate these cytosine residues, the transcription factors don't bind, and so RNA polymerase also doesn't bind. And so there's no transcription. If you see what I've said, here, transcription is initiated by RNA polymerase binding to promoters. We know this. Promoters are rich in the base cytosine. Adding a methyl group to cytosine within promoters prevents RNA polymerase and transcription factors from binding, resulting in gene silencing. Demethylation, on the other hand, activates gene expression. The enzyme which methylates promoters is called DNA methyltransferase. The name tells you what it does, right? It transfers the methyl group to DNA, or the short form is DNMTs. Good. So someone asked, so histone acetylation increases gene expression, but DNA methylation decreases gene expression. Yes. And histone deacetylation will decrease gene expression, and DNA demethylation will increase gene expression. So it's the opposite. Okay. So now you might think why kind of you guys want to learn this as medical students, but because this is a major field in research, especially cancer research. So if you have kind of the paradigm I explained to you earlier about how normal cells have two sets of genes, tumor suppressor genes, which are expressed, and oncogenes, which are cancer promoting, so they're silenced. In a cancer cell, what happens is that you have histone acetylation changes. You have abnormal histone acetylation and abnormal histone deacetylation. This results in expression of these cancer promoting oncogenes. This results in decreased expression of tumor suppressor genes. And this decreases gene expression of metastasis reducing genes. And this decreases DNA repair genes. All of these different gene expression changes, which can be explained by abnormal histone acetylation slash deacetylation, all of them have been implicated in breast cancer metastases. And so if you know these mechanisms, what researchers can do is they can develop drugs which inhibit or which decrease the acetylation of these genes, or they decrease the deacetylation of these genes, so they can normalize the gene expression. The methylation is happening on the promoter itself, yes. Okay. What is EMT? You don't need to know that. EMT is epithelial to mesenchymal transition. It allows cancer cells to kind of invade connective tissues and cause metastases. Okay. So this is kind of just me giving you some extra information to highlight why this topic is important for you guys to know. So this is a normal gene, is a normal cell. You see, this is the promoter of a gene. This is exon. These are exons here. This is the promoter of a gene. And these are all cytosine residues, these spikes here. And so this gene is a tumor suppressor gene in a normal cell, and it's expressed. And it's in an open or euchromatin conformation because its histones are acetylated. In a cancer cell, what happens is you methylate these. You can see all of these methylated cytosine residues. So in cancer cells, this gene, this tumor suppressor gene, is hypermethylated, and it goes into kind of a closed chromatin conformation. which basically means is histones get deacetylated. And so this gene gets turned off. And if this was a tumor suppressor gene, then you can guess that that cell has higher risk of becoming cancerous, right? So we're done with epigenetics. So this general organization that I gave you at the beginning, that you have transcriptional regulation, which is two types. Epigenetics. Epigenetics is histone acetylation, DNA methylation slash demethylation. The other part of transcriptional regulation is called DNA binding protein or transcription factors. Okay. What is CPG? You don't need to know that either, basically. It's not important for you guys to know. CPG is basically cytosine attached to a guanine. Okay. So CPG, cytosine guanine dinucleotide. So now from now on, I'm going to refer to DNA binding proteins as transcription factors. Okay. So as the name suggests, transcription factors are proteins which bind to DNA and they either increase or decrease transcription of a certain gene. Before we move on to the second section, can you rediscuss the tumor part? The tumor part was to kind of allow you to tie in these concepts together. What you should know from what I've just discussed is what DNA methylation slash demethylation do and what histone acetylation slash deacetylation do. There's specific alterations in the context of certain diseases or cancer is not really that important, but you could get asked questions in a in the context of cancer. So they could ask you that, for example, they could give you an example of a guy with colorectal cancer, and they could tell you that in that patient, his cancer cells show DNA methylation of a certain tumor suppressor gene. What will that do? The mechanism they're asking you is very simple. What does DNA methylation do? That's what the question is. But they're asking you it in the context of a cancer cell. So the cancer becomes a bit of a distraction, right? So that's why I explained it to you. So you can just get an idea of how these things can be tested in questions in the context of cancer. Okay. So transcription factors are proteins. This is important. The transcription factors are proteins which bind to DNA, and they either increase gene expression or decrease gene expression. Transcription factors are of two types, general or specific. General transcription factors bind to promoters to facilitate RNA polymerase binding. Okay. So general transcription factors bind to promoters to facilitate RNA polymerase binding. And this is associated with a basal transcription level. Let me give you an example, right? For your body to deal with glucose circulating in the blood, for your body to deal with it, you need a hormone called insulin. And glucose is constantly circulating in your blood, right? At a very steady, within a set limit, within the homeostatic limit. For that, you need a very small amount of insulin, which is constantly being synthesized and released by your pancreas. This expression of insulin is mediated by general transcription factors. Okay, so general transcription factors bind to promoters and they're associated with a steady basal low level of transcription. But then suppose I eat a high carbohydrate meal and my blood glucose shoots up. Now I need a lot of insulin to deal with that blood glucose. And so my gene expression of insulin needs to go very high. So in that context, I need things called specific transcription factors. These specific transcription factors are also proteins, but they don't bind promoters. They bind areas of DNA called response elements or REs, okay? Response elements. And the way that they bind them is that they use specific parts called zinc fingers. And so you can think of a transcription factor like a fist. When it gets activated, it opens up. And these fingers, these zinc fingers, allow it to bind the response element. What happens when this specific transcription factor binds the response element? That it causes a massive increase in insulin gene expression. And so now my body can release a lot of insulin to deal with the nutrient load I just ate. Okay, so specific transcription factors are when you need a lot of gene expression. So this is kind of a schematic of what I just said. This is the transcribed region. This is the gene. This is the promoter of the gene. This is where general transcription factors will bind, and it'll be associated with a basal transcription level. Okay. Then you have areas which are very, very far away from the DNA. You go 1,000 base pairs back and you reach a region called a response element. A response element which activates gene expression is called an enhancer. Okay. So they're response element. You can see the RE here. They're both all response elements. But because they activate gene expression, they're called enhancers. Specific transcription factors will bind these enhancers by which mechanisms? Through zinc fingers, and this is associated with an increased gene expression. How do they do this? Now, are basal transcription levels steady slash low? Yes. Basal transcription level are low, very low. Because your body, I gave you an example of insulin, right? Your body needs a low amount of insulin constantly. And so general transcription factors allow you to synthesize a very steady slash low amount of insulin. When you need a lot of insulin, when you eat a high carbohydrate meal, for instance, your body needs specific transcription factors. Okay. So now you know where transcription factors bind, you know how they bind through zinc fingers, and you know that they increase transcription. There's one more thing you need to know is that when these specific transcription factors bind, right? So you've opened the zinc fingers and you've bound the enhancer. What will happen is that the DNA will bend. Okay. So you have a promoter here. You have to kind of use your imagination here. You have a promoter, and very, very far away, you have an enhancer which has been bound by transcription factor. What will happen is that this DNA will bend and this enhancer will come very, very close to the promoter because the promoter is here, the enhancer is here, right? The DNA will kind of bend like this, and this will give a very comfortable seat for RNA polymerase to bind to the promoter. You can think about it like that, okay? So this bending is to provide a comfortable seat for RNA polymerase, kind of recognize that promoter and say, this is the gene that I need to transcribe lots of mRNA. And so this will increase gene expression, okay? Can specific transcription factors inhibit gene expression? Yes, they can. In that context, they will be called repressors. We'll get into some of the terminology here, but for a start, you need to kind of be oriented with what specific transcription factors do when they're increasing gene expression. That's why I started with this kind of unidimensional view on transcription factors, that they increase gene expression. They don't always increase gene expression, but that's this thing you should start off with because it's easier to understand, all right? So this is the kind of a schematic representation. You have the gene here, you have the promoter here. These general transcription factors are binding this promoter, and they're allowing RNA polymerase to bind, but the seat is not as comfortable. And so you have very steady slash low levels of transcription. I gave you the example of insulin. Far, far away from the promoter, you have a region called an enhancer. This is a type of response element, okay? This is specifically a response element that increases transcription. You have a specific transcription factor, which is called an enhancer, okay? This specific transcription factors bind the enhancer, and this kind of causes the DNA to bend by recruiting proteins called DNA bending proteins, all right? So these DNA bending proteins, they are recruited to the DNA, and they cause the DNA to bend. This creates a very comfortable seat for RNA polymerase to promote transcription, okay? Does this make sense so far? Now we can ask questions. Okay, so general transcription factors bind to promoters, and specific transcription factors bind to enhancers, bind to response elements. Absolutely, yes, exactly. And this binding of general transcription factors to promoters is associated with a basal slash steady slash low level of gene expression. The binding of specific transcription factors to response elements like enhancers will be associated with a massive, massive increase in gene expression. Are enhancers response elements? Yes. So let me kind of make this clear for you. This is a diagram from Lippincott. It's kind of showing you the same thing, right? You have RNA polymerase binding to the promoter. You have general transcription factors, i.e. basal transcription factors. You have specific transcription factors binding to enhancers, causing the DNA to bend, recruits RNA polymerase. But let me kind of orient you guys with the terminology, all right? Based on their function, So you have response elements as like an umbrella term, right? Based on their function, they're divided, response elements are divided into enhancers if they increase transcription. If they decrease transcription, they're called silencers. And based on their function, specific transcription factors are divided into activators. If specific transcription factors bind to enhancers, they're called activators. If specific transcription factors bind to silencers, they're called repressors, all right? So this is what you need to know here. This is a response element which activates gene expression, so it's called an enhancer. If this is an enhancer, the specific transcription factor, which will bind it through zinc fingers, will be called an activator, okay? If this response element decreases gene expression, it's called a silencer. If this is a silencer, the specific transcription factor, which will bind the silencer, is called a repressor. Insulator is an interesting one. You have an enhancer here, you have a response element, it's bound to an activator. So your idea would be to increase gene expression. An insulator is a regulatory element or a response element which kind of interferes with this mechanism. So it interferes with the enhancer slash activator mediated increase in gene expression. Okay. So an insulator, the name kind of gives you insight into what it does. It kind of insulates the activity of an enhancer slash an activator. Okay. The insulator thing is not really, I don't think, that important. I don't remember Dr. Aljada kind of testing you on that, but you should know this kind of general classification of response elements and of specific transcription factors. We are strictly talking about eukaryotic cells here, right? Yes, we are. Yes. Prokaryotic cells have a very, very different mechanism of gene expression, and they're regulated a bit differently as well. They're regulated through things called operons. Operons are a cluster of genes associated with one promoter. Whether or not that gene is active or that promoter is active depends on environmental conditions. Okay, so that's kind of how prokaryotic genes are regulated. This is strictly for eukaryotes, for humans, right? So we've discussed what transcription factors are, what two types of transcription factors are. We've discussed where they bind, how they bind by zinc fingers. We've discussed the fact that they cause DNA bending. And we've discussed that they can either increase gene expression or decrease gene expression, depending on what they are, silencers slash repressors or enhancers slash activators. Does this make sense to everyone so far? Before we get into this diagram? Yes? Okay, that's good. Okay, so this diagram is showing you an example of how, can you repeat the silencer part, please? Sure. So do you understand what an activator is? Or activator slash response element? Exactly. So what is an activator? An activator is a specific transcription factor which binds to an enhancer. An enhancer is a type of response element which increases gene expression. The opposite type, i.e. a response element which decreases gene expression, that's called a silencer. Okay, so it's the opposite of an enhancer, essentially. Do you get that now? Okay, so the last block that you guys will take in your first year is endocrinology. In endocrinology, you learn about the mechanism of action of different hormones. Hormones are essentially proteins or lipids which your body releases into the blood in certain situations to maintain homeostasis. Okay? If you can understand this slide, you will understand how hormones work. So let me give you an example, right? Let's go back to kind of the drawing board. Okay. Let's suppose you have you just ate a meal. Blood glucose goes up. Okay? Blood glucose increases. Your body will release a hormone called insulin. Insulin, because it's while your body wants to maintain homeostasis, logically, your body releases insulin to bring the blood glucose back down. So insulin will activate pathways. Question. Okay, so insulin will activate pathways which use up glucose to decrease the glucose, all right? On the other hand, if your body is starving or you're in fasting conditions, you have decreased blood glucose. So your body releases hormones called glucagon or cortisol, okay? Cortisol. These two hormones you need to know, glucagon or cortisol. These will increase blood glucose. Now, this is what you learn in endocrinology, but this is the big picture here. How will they increase blood glucose? Logically speaking, it should make sense that they will activate pathways which produce glucose. So this pathway here that we're looking at is called gluconeogenesis. Gluconeogenesis is the production of new glucose. Okay? This is the production of new glucose. When would that be active? In fasting conditions. So you have amino acids which you're getting from muscle protein breakdown. When you're starving, you have muscle wasting, right? Your muscles are kind of decreasing in size, and those proteins are being broken down into amino acids. Those amino acids are being funneled into a compound called OAA or oxaloacetate. Oxaloacetate gets converted to the molecule called PEP, phosphoenol pyruvate, that gets made into glucose. Glucose goes out into the blood, and this maintains the blood glucose. So that's homeostasis. That's the mechanism of action of glucagon and cortisol. But how do they achieve this action? No, you don't need to know these specific names here. This is kind of just to orient you with this diagram because people find this diagram quite confusing. You need to know the one name which I'll tell you, okay? You don't need to know these names here, PEP or OAA. You don't need to know that. So how do glucagon and cortisol increase gluconeogenesis? Okay, they increase gene expression of an enzyme called PEPCK. You need to know this name. You only need to know this name. PEPCK, PEP carboxykinase. This enzyme mediates the rate limiting step of gluconeogenesis. When you have a lot of this enzyme, this gets converted to PEP, PEP to glucose, and you have a lot of glucose circulating in the blood to maintain blood glucose. Okay, now how do they do this? Cortisol is a steroid hormone, okay? Cortisol is a lipid-soluble hormone. Because it's lipid-soluble, that means it can easily pass through the cell membrane because the cell membrane is hydrophobic. The interior of the cell membrane is hydrophobic. That means anything which is lipid-soluble can pass through the cell membrane. Cortisol binds to its receptor. This slanted thing here, this V-shaped thing, it's the cortisol receptor. So now imagine this. This is the cortisol receptor, it's shut off. Cortisol comes in and binds to it, and this receptor kind of opens up, and it has these zinc fingers sticking out. So this receptor is an example of a specific transcription factor. Okay, so the cortisol receptor is an intracellular receptor and is a specific transcription factor. This cortisol receptor complex with zinc fingers goes to a response element called GRE, glucocorticoid response element. Cortisol is also called glucocorticoid, and this is a response element. And this response element would be an enhancer. When this cortisol receptor binds to GRE, the DNA bends, RNA polymerase transcribes PEPCK by a lot, and you have increased expression of PEPCK, and you have gluconeogenesis. Okay? Someone asked Muhammad, is the cortisol receptor an enhancer or a silencer? Okay, so let me kind of just get the terminology here. Specific transcription factor binds to response element. Yes, it's an activator, exactly. So a specific transcription factor is a protein which binds to a response element. Response element is a part of the DNA. Okay, let me show you here. Response element, this is the response element. It's a part of the DNA. Now, a response element can be an enhancer. It can be a silencer. Okay, so you have the response element, which is the DNA. It's not this transcription factor. The DNA can be an enhancer or a silencer. The transcription factor, if it binds an enhancer, it's called an activator. If it binds a silencer, it's called a repressor. Okay, so enhancer, silencer are response elements. Activator, repressor are specific transcription factors. Okay, now, so the cortisol receptor is a specific transcription factor. Because it's binding an enhancer, As Bilal said, it would be an activator. Okay. Do you understand that, Mohammed?
Oh yeah, thank you, thank you.
Okay, good. So this is the action of the cortisol receptor. And you should understand this because every single hormone that you study in endocrinology, which is lipid soluble, works through this mechanism. And this explains a certain difference between lipid soluble hormones and water soluble hormones. Let me give you an example. Let me kind of elaborate here. Glucagon is a water soluble hormone, right? Glucagon is water soluble, which means it can't cross the cell membrane, which means its receptor has to be at the cell surface. When it activates this receptor, this activates a G protein called GS. You don't need to know that. This increases cyclic AMP. Cyclic AMP activates protein kinase A. Okay? Protein kinase A, any kinase enzyme that you see in biochemistry, causes the addition of a phosphate group or phosphorylation. When protein kinase A phosphorylates Krebs, it activates it. Krebs is cyclic AMP response element binding protein. Okay? Krebs. Protein kinase A phosphorylates Krebs and activates it. Krebs is lying dormant or inactive in the cytoplasm until glucagon binds its receptor and PKA gets activated. Protein kinase A gets activated. Krebs then opens up with its zinc fingers. It goes to the DNA. It goes to CRE, cyclic AMP response element, an enhancer. So Krebs would be an activator. When this happens, Krebs causes the DNA to bend, and RNA polymerase transcribes the gene PEP-CK. PEP-CK gene is expressed. It's increased gene expression. You have increased gluconeogenesis, and so you maintain blood glucose. Now, that explains why steroid hormones have, you study this, steroid hormones take longer to act than water soluble hormones because steroid hormones work through gene expression only. The water soluble hormones work through many other mechanisms. This PKA will go and modify many enzymes in the cytoplasm, which are already there. Steroid hormones can't do that because they don't have membrane bound receptors. And so they don't have a signaling pathway. They go directly into the cell and they bind to their receptors, which are specific transcription factors, which means their zinc fingers. When they get activated, these zinc fingers bind to DNA, cause modified gene expression. This modification takes time. Hence, steroid hormones are slow to act. And there's many, many steroid hormones in the body. Testosterone is a steroid hormone. Estrogen is a steroid hormone. Aldosterone is a steroid hormone. All of them work through this same mechanism of passing through the cell membrane, binding to their intracellular receptor, activating zinc fingers, modifying gene expression. Cortisol works with glucagon, or are they different processes? That's a good question. Um, Cortisol is released by your adrenal gland mainly under stress circumstances. Okay, so when you're exercising, for example, because you're exercising, you need glucose. It's a stress response. Cortisol is active during stress. Glucagon is mainly active during fasting. Okay, but the way they achieve uh, the way they both activate gluconeogenesis or the production of glucose is the same. You get my point? So they're active in different circumstances. But the way they impact glucose metabolism is the same, is by modifying the PEP-CK gene expression. Does that make sense? So they kind of have a synergistic effect. So they don't need one another. They can work separately. Yes, exactly. Yes. So they're not both working at the same time. Actually, on the hepatocyte, glucagon has the predominant effect. Okay? In fasting conditions, glucagon is mainly the one driving gluconeogenesis. But cortisol can do that. Can you explain the last Krebs and cortisol part? I didn't focus. Sorry. Okay, I will. Is glucagon also a specific transcription factor? No. The specific transcription factor is the protein which binds to DNA. Glucagon isn't passing through the membrane, is it? So glucagon is only binding to its receptor. Krebs is an example of a specific transcription factor with zinc fingers. Do we need to know the motif versus domain? What do you mean by that, Mubarak? So they're both activators which activates separate enhancers. Yes, exactly, Saad. Yes, it's in the slide. Can you explain the glucagon part again? Sure, I will. I'll go through it again. So, the only motif versus domain part that you need to know if you're referring to this one is the way they bind to the response element. They bind through zinc fingers. Okay, there's a lot of stuff in your slide about cis regulatory elements versus trans regulatory elements and stuff. Not important. Okay, this is what's important. If you can understand this, it's enough. Okay, so let's discuss. Sidra, I think, asked, and someone else asked about repeating this. Okay, cortisol and glucagon, we discussed on the whiteboard, both increase glucose. Okay, and the way they increase glucose is by increasing glucose production by activating the expression of this enzyme, PepCK. The way they achieve this is different. Cortisol is a steroid hormone, is a lipid soluble hormone, passes directly through the cell membrane, activates its intracellular receptor. The intracellular receptor and cortisol complex, they both go into the nucleus. The intracellular receptor is a specific transcription factor which activates the zinc fingers. It kind of spreads them out, binds to the DNA, causes bending, and increases gene expression. Okay, that's cortisol. The cortisol is the hormone. The receptor is the specific transcription factor. Glucagon is totally different because glucagon is not lipid soluble. It doesn't pass through the cell membrane. It only binds through this. It doesn't pass through the cell membrane. It binds to a surface receptor. It binds to its receptor, activates cyclic AMP. You need to know that. Cyclic AMP activates protein kinase A. I remember this as AMP activates A, right? So A goes with A. Cyclic GMP activates protein kinase G. Okay, so AMP goes with PKA. When PKA gets activated, it it kind of phosphorylates things. Any kinase enzyme phosphorylates different enzymes, okay, different proteins. Depending on what protein it is, it's either active or inactive. So PKA finds PrEB lying around in the cytoplasm inactive. PrEB is cyclic AMP response element binding protein, all right? And it's lying around inactive in the cytoplasm. PKA phosphorylates PrEB, activates it. PrEB spreads out its zinc fingers, goes to the DNA, binds to CRE, cyclic AMP response element, which is an enhancer. So PrEB would be an activator. This causes DNA bending. The PepCK gene gets transcribed and translated, and gluconeogenesis happens. Okay? Any questions? Okay. Rida, also, can you, does it make sense to you? Yes? Okay, good. So we move on now. This is kind of summarizing what we just said. Um, this is transcriptional regulation. You kind of remember that membrane-associated receptor is an example of an enhancer. No, enhancer is the response element on the DNA. The membrane-associated receptor is simply responsible for binding glucagon and transducing its signal to the cell. Okay, so there's a few questions here. I have a question. Cyclic AMP usually causes phosphorylation through kinases and hence activation of a protein. I mean, generally in cell signaling pathways. This is a bit more complex. PKA, protein kinase A, phosphorylates different proteins. Depending on what pathway they relate to, they're either activated or inactivated. So let me give you an example. Cyclic PKA will phosphorylate PrEB and it will activate it because PrEB is involved in gluconeogenesis. PKA will also phosphorylate enzymes which are in glycolysis. Those enzymes will be inactivated. So depending on what pathway we're talking about, the cyclic AMP PKA pathway has different effects, inactivation or activation. So now Rida asked, so regulation essentially, yes, exactly. So the pathways which are activated by insulin are inactivated by glucagon. The pathways which are activated by glucagon are inactivated by insulin. I know you told us not to focus on cis-acting factors, but aren't they response elements? Yes, exactly. So it's just some terminology you don't need to know. Okay, so essentially cis regulatory elements are sequences of DNA which regulate gene expression of a gene on the same chromosome. All right. So they're present on the same chromosome. Enhancers, promoters, silencers, insulators are all present on the same chromosome as the gene they regulate. So they're all examples of cis regulatory elements. Transcription factors, they don't belong to that chromosome. So they would be examples of trans regulatory elements. Okay. So again, it just gets muddled up with terminology. And I don't want you guys to get muddled up with terminology. I don't want you guys to understand the concept. Okay. Good. So this is transcriptional regulation in a nutshell. There's two types, epigenetics and transcription factors. Epigenetics, again, two types, histone acetylation. Histone, the enzyme is histone acetylase or histone deacetylase, HDAX. Histone acetylation of lysine residues on histone tails. This affects DNA histone electrostatic attractions. Acetylation equals activation. Deacetylation equals deactivation. That's how you can remember it, right? DNA methylation is mediated by enzymes called DNMTs, DNA methyltransferases. They methylate promoter cytosine residues. This methylation causes gene silencing. This by controlling the binding of how methylation causes gene silencing is that it prevents RNA polymerase and transcription factors from binding to the promoter. What transcription factors specifically? General transcription factors. Okay. This is all on epigenetics. Transcription factors are two types. General, they bind promoters associated with basal/low/steady transcription. Specific, they bind response elements. The response element can be an enhancer, it can be a silencer. So the specific transcription factor can be an activator or can be a repressor. The activators cause DNA bending and increase gene expression. The one example you need to know right now of this is PepCK regulation by glucagon and cortisol. But every single hormone you study in endocrinology and in biochemistry, insulin works the same way, aldosterone works the same way, every single hormone in your body works the same way. So that's why studying that whole pathway of how hormones influence gene expression is very, very important for you guys to understand. Okay? So this is all on transcriptional regulation. Any questions here so far? We're at 8 o'clock. Okay. How do residues work? Okay, so you know how DNA is made up of adenine, guanine, cytosine, thymine, right? So these are different nucleotides. Each of them is called a residue. So I would say it's terminology. I would say the thymine residue of DNA or the cytosine residue of that promoter. Okay. Creb is an activating transcription factor. Yes, it would be an activator because it's binding and enhances. Exactly. Cortisol and Creb are both transcription factors because they bind through zinc fingers. The cortisol receptor is and Creb are transcription factors. Cortisol itself is the hormone which activates the receptor. The receptor is the transcription factor. Okay? The receptor is the one with zinc fingers. The Creb is the one with zinc fingers. Yes. Okay. So we've had a lecture about 45 minutes. I can finish within the hour. Any other questions? Okay. So we'll go on to translational regulation. This is very, very simple. So translational regulation. Now you have the mRNA, you have the DNA, you had transcriptional regulation. That gene got transcribed into mRNA. That mRNA gets capped at the 5' end by 7-methylguanosine cap. It gets polyadenylated at the 3' end, gets spliced, moves out into the cytoplasm as mature mRNA. Then you can still regulate that. You can regulate it by degrading it, okay? So mRNA transcripts can be degraded by nuclease enzymes, so they cannot be translated. Their stability, the stability of each mRNA molecule is determined by the sequence in the 3' untranslated region. You don't need to know that, these are just details, but you should know the entire concept, which is that mRNA stability can be controlled, and this is a form of post-transcriptional regulation, okay? You can inhibit the mRNA from being translated. So mRNA is in the cytoplasm. It can be bound to ribosomes. It's not being degraded, but you can inhibit translation. And we'll discuss that in the next slide. The last step, the last component of translational regulation is RNA interference. There are two types. small interfering mRNAs or siRNAs or microRNAs. I need you to focus on microRNAs, okay? They bind mRNA, so both of them siRNAs or miRNAs, they both bind their target mRNA, their target messenger RNA, and they can either inhibit it or degrade it. Okay, so these two pathways, they bind the mRNA, inhibit it, or degrade it. This is called RNA interference. The main thing is microRNAs. Okay, so let me kind of explain to you again on the drawing board here. So, you have your gut, your intestine, iron from food you eat, mainly meat, is found in the Fe2 plus state. It goes into the blood. Iron is bound to transferrin in the blood. This protein called transferrin is in the blood. It binds iron. When iron gets into cells, is bound to a protein called ferritin. Okay? Yeah, exactly. So I'm just kind of making it simple for you. So you took iron metabolism. Okay, that's good to hear. So you know transferrin and ferritin. Okay, that's very good. Yeah. Okay. So the reason why, so when you took iron metabolism, I think you guys would have noticed that iron is never let free in the body. You never let iron free in the body. You always bind it to proteins. The reason for that is that iron can generate free radicals. It can generate reactive oxygen species, and these damage your cells, they damage DNA, they damage proteins. So you can never let iron free in the body. You always bind it to protein. The only thing which concerns me, about this lecture, is that you know this. Okay? You have iron bound to ferritin in the cell. Why is this important? Because when you have a lot of iron in the cell, you'll need a lot of ferritin. Isn't it Fe3 plus in the body or in the system? It's Fe2 plus. Okay? Yeah. So when you have a lot of iron in the body, you need a lot of ferritin in the cell. When you have a low amount of iron in the cell, you need a low amount of ferritin in the cell. So then logically, it should make sense that the amount of iron in the cell is controlling the amount of ferritin you have. That's why when you have iron deficiency, ferritin is way down because you don't have iron, so you don't need ferritin. So the amount of iron in the cell is controlling expression of ferritin, but it's not controlling expression of ferritin at the transcriptional level, not through epigenetics, not through transcription factors. It's controlling it at the translational level, and which is what we'll look at now. So this is the mRNA for ferritin. It's roaming around in the cytoplasm, ready to be translated. You have the polyadenylated tail here. You can see that. You have the start codon, you have the stop codon. In the 5' untranslated region, where the ribosome will bind, there's a base sequence called an iron response element. This is different from the other response elements we took. The response elements we took in transcription were on DNA. This is not on DNA, this is on mRNA, okay? Iron response element or IRE. When you have low iron, you have no iron in the cell, this IRE is bound to a protein called IREB, IRE binding protein, okay? This IREB, when it binds to IRE, it prevents the ribosome from binding to the mRNA. Hence, it stops translation, okay? So low iron equals IREB binding to IRE. That prevents ribosome from translating. That prevents ferritin production, which is what I've said here. Low iron inside the cell, low intracellular iron concentration indicates that ferritin is not needed. So its translation is suppressed by IREB binding IRE. On the other hand, if you have iron inside the cell, which means you need ferritin, these iron molecules bind to IREB and they remove it from the IRE. This means that the ribosome can bind to the ferritin mRNA at the 5' UTR and translate it into the ferritin protein. Okay? So low iron equals low ferritin through this mechanism. What do we normally find in the 5' untranslated part? You find the ribosome binding site. Okay? That's what's important. It's not translated, so it's not translated into a protein. It's important for the ribosome to recognize the mRNA And so hence, because this IRAB is binding here, the ribosome can't bind. You have a question? So Fe is included in RNA interference since it degrades. I don't understand that question. The 7-methylguanosine, yes, Bilal. Sidra, can you explain your question? So, okay, I think I understand your question actually. So she's asking if ferritin, if this iron regulation is included in RNA interference. Yes. miRNA either inhibits or degrades. No, this is not RNA interference. RNA interference is interference by RNA. So microRNAs are actual RNA sequences which are interfering with translation. So that's why it's called RNA interference, because it's RNAs which are interfering with translation. These are proteins which are interfering with translation. Okay? So it's not RNA interference. Okay, so this, I think this kind of makes sense to everyone. This is another diagram. This is the 5' end of mRNA. This is the 3' end. This is the IRE in the 5' UTR. IRE binding protein bound. So with low iron, there's no translation. When iron comes in, it removes this IREB or IREB. IRE is free. Ribosomes can bind and you can translate and produce ferritin. Okay? If ferritin is needed, why do we stop it from translation and not transcription? Because each gene has its own way of regulation. Okay? And ferritin is an location of expression. So ferritin is not expressed in all the cells in the body. It's expressed in certain cells in the body, and those specific cells in the body have specific ways of regulating it. Okay? So let's talk about RNA interference. Now, this is a complex diagram. It's from a journal, but you don't need to know it. All you need to know is that you have 3 mRNA and then you have mature mRNA, right? 3 mRNA has not gone capping, has not undergone splicing, has not undergone polyadenylation. Just like that, you have 3 microRNA or pre-miRNA. This pre-miRNA runs into an enzyme called dicer. Dicer is an RNase enzyme. This cleaves the pre-mRNA, pre-microRNA, and this shortens it to form mature microRNA. These mature microRNA then get associated with these proteins called Argonaute proteins, which form an RNA-induced silencing complex, or RIS. This microRNA, when it associates with this RIS, it forms an RNA RIS complex. This RNA RIS complex has a specific mRNA it recognizes. When it recognizes it, it cleaves it and degrades it. Okay? That's RNA interference in a nutshell. The other part is small interfering RNAs. They're synthesized by precursor called short hairpin RNAs. They get also cleaved by dicer or RNase to form mature small interfering RNAs. They complex with RIS proteins or Argonaute proteins to form an RNA RIS complex. These RNA RIS complexes, they bind to a specific mRNA. When they bind to it, they're degraded. If this binding to the target mRNA is an incomplete match, so it's not all complementary, they inhibit it from translation. Okay? So if it's a complete match or a perfect match, it's degradation. If it's an imperfect match, it's inhibition. Does that make sense? It's a complex diagram, but makes sense to everyone? Any questions?
You repeat the last part, the imperfect and the inhibition.
Okay, sure. So I think both of us understood and what happens until microRNAs and small interfering RNAs, they complex with the RIS protein and they form this complex. When they form this complex, they have a specific mRNA that they're complementary for or that they want to target. Let's say that mRNA is the mRNA of PepsiK, let's say. It's not, but let's give you an example. Just to give you an example. Let's say they find the mRNA of PepsiK and it's not all complementary. Those base strands that they're binding to and they're linking to non-covalently, they're not all complementary strands. So if it's an imperfect match. If it's an imperfect match, they inhibit it from translation. In other words, they prevent ribosome from binding to the mRNA and translating it. If it's a perfect match, then they degrade it by enzymatic cleavage. Okay? So there's two options depending on perfect and perfect.
Okay, thank you.
Okay. Any other questions? Mustafa, did you understand? Okay.
Wait, one more question.
Yeah.
How does it being imperfect not allow for translation?
I'm not sure, to be honest. I'll have to look into the mechanism of that. But by just the fact, intuitively, it should make sense that if something is bound to mRNA, ribosomes themselves cannot bind, right? The specific
Yes.
interactions which go into that, I don't really know, but kind of intuitively it makes sense if you think about it, yeah? Because ribosomes need to bind and go through the entire sequence. If there's these proteins and RIS and RNA complex, which is in the way, they won't be able to translate it. But I'll have
Yes.
to kind of search it up and get back to you. Okay?
Yes, thank you.
Okay, so this is kind of the text. I've summarized it. It's RNA interference and introduction. It includes small interfering RNAs. And why not degrade all the semi-complementary mRNA? I'm not sure. Yeah. I'm not sure specifically what enzymes are involved and stuff. I'm not really sure about that. After the mRNA is cleaved, it will not be able to express into proteins. Yes, exactly. Because you require a complete mRNA with a 5' UTR and methylguanidine cap. Those won't be there if the mRNA is all broken up, right? So they won't be recognized by ribosome. So RNA interference includes small interfering RNAs or microRNAs. These both of them have precursors. For the precursor of microRNA is called pre-M microRNA. This runs into an enzyme called dicer, which matures them by kind of shaving off their unnecessary parts. Why does it look like a double helix? It's just a diagram, man. Like, yeah, it's just to kind of explain a concept. And it's not a double helix, it's a single helix actually. And this part is the part which is bound to the mRNA. Okay, you can see this interaction here between the RNA, the microRNA, and the target mRNA. Otherwise, it's a single strand. Okay, so these microRNAs complex to Argonaute proteins to form a RIS RNA complex. The RIS complex binds to complementary mRNA. A perfect match traffics that mRNA for degradation. An imperfect match inhibits that mRNA from being translated. Okay, microRNAs, if you think about it now, microRNAs give you a mechanism of regulating gene expression without specifically messing with the nucleotide sequence of the gene itself. So microRNAs are considered the third and final component of epigenetic regulation. Okay, so epigenetics has three components in general: histone modifications, histone tail modification, acetylation, deacetylation. There can be methylation as well, but that's not important. Acetylation, deacetylation, methylation/demethylation of promoters, and microRNA mediated RNA interference. These three are the three components of epigenetics. Okay. Now, I kind of told you that I was going to link these things to cancer. So what you can do is look at the genome of a cancer cell. Let's say a colon cell, a cell in your liver, for example, which has turned cancerous, and compare it with the gene expression of a normal liver cell. What you'll notice is that the cancer cell has lower tumor suppressor mRNAs. So the tumor suppressor genes are suppressed, and it has higher oncogenic miRNAs. So the M microRNAs, which are allowing expression of oncogenes, are lower, and so oncogenes are being expressed. The microRNAs of tumor suppressor genes are not allowing those genes to be expressed. So there's an imbalance between gene expression, again, between pro-cancer and anti-cancer pathways. And so you can again target that with these molecules, miRNA mimics, small molecules, sponges, antagomere, etc., etc. So all of these mechanisms that we just looked at are actually very relevant from a oncology perspective. You'll kind of find these drugs being used in KFSH and other hospitals as well. Okay, so this is the summary. Gene expression divided into transcriptional or translational. Transcriptional is mediated by transcriptional factors, general or specific, or epigenetics like histone modifications, or promoter methylations like demethylation. Translational regulation includes either degrading mRNA or inhibiting it from translation, which can be done via microRNAs or IREBs in the case of ferritin translation. These are some questions. I'm done with the lecture. Anyone have any questions from the lecture first? No? Okay. Okay, good. Do you want to go through the questions or would you rather do them on your own or?
Wait, can I just ask one question?
Sure.
Sorry. In the cortisol and glucagon, you said they kind of work during different times, right? Just again, one more time. What do they each one? What does each one do exactly?
Okay. Cortisol is a stress hormone, okay? So when you're stressed, your body releases cortisol. Cortisol's job and glucagon's job overlap. Both of them are maintaining blood glucose through gluconeogenesis. Glucagon is active during fasting conditions, okay? All the time. Cortisol is not always active. It's a different hormone with different regulation, with different circumstances in which it increases and decreases. But regarding gluconeogenesis, their phenotypic effect is the same. They increase glucose in the blood. How they do this is by increasing PepCK gene expression. How they do that is by binding to GRE in case of cortisol, and by binding to CREB and activating CREB and binding to CRE in case of glucagon. So the mechanism is slightly different, but it's the same principle. Okay?
Yes, thank you.
Yeah. Go through the questions, go through them, okay? Cortisol level when you stress. Fine. Okay, so Asma asked, can you please explain that what happens during a perfect match of RNA risk complex? Okay, so you have the RNA risk complex, right? It binds to its mRNA, messenger RNA that it wants to bind to. It's bound to it. If it's a complementary match, it degrades it. If it's an imperfect match, it inhibits it from translation. It just keeps bound to it. These specific questions, I don't think they would ask you that in an exam. Like, for example, it's an incomplete match, what's the next step or what happens next? That's that's minute details, okay? But you need to know the concept of RNA interference and you need to know that they degrade mRNA. Why did you say that mRNA is an epigenetic modification? That's a good question. Epigenetics is defined by regulation of gene expression without altering nucleotide sequence, without messing up with the sequence of a gene with mutations and stuff, and regulating gene expression. That's called epigenetics. So epigenetics includes DNA methylation, histone acetylation, and microRNAs. Okay? Specific transcription factors include activators and repressors, right? Yes. And the response elements they bind to, respectively, are enhancers and silencers. Okay. So, okay, if you don't mind, can you please go through the mRNA, siRNA thing once again? Sure, I'll go through it once again, but I would like you to do these questions first, okay? So, a 45-year-old man or patient with poorly controlled type 2 diabetes presents with persistently elevated fasting blood glucose levels. His doctor tells him that gluconeogenesis is a key process contributing to his elevated blood glucose. Which of the following transcription factors primarily regulates the expression of key enzymes involved in gluconeogenesis? Which of these transcription factors? You can type in the chat, unmute your mic. B. Yes, B. Cyclic AMP response element binding protein. What's it activated by? Glucagon. Exactly, through protein kinase A, and it binds to CRE. Okay, that's good. So, a 50-year-old male patient is diagnosed with hypertension. Further investigation revealed an upregulation of angiotensin 1 receptor type 1 on vascular smooth muscle cells. This is a distraction, not important. Research suggests that a specific microRNA is involved in the post-transcriptional regulation of this receptor. Which of the following best describes how microRNAs typically regulate gene expression? Yes, microRNAs bind to the 3' UTR of target mRNAs. Um, I think I've copied the question wrong, but yeah, so yeah, it's correct. So microRNAs bind to the 3' UTR of target mRNAs, inhibiting translation or promoting mRNA degradation. So Reem, this is all you need to know about microRNAs actually. That's this mechanism of action. Regarding whether it's a perfect match or imperfect match, and what are the specific mechanisms involved, that's not important. Just know this sentence, okay? MicroRNAs, form of RNA interference. They bind to target mRNAs, inhibiting translation or promoting mRNA degradation. Also know that the enzyme dicer is responsible for their maturation. Okay? This is the last question. A 60-year-old patient is diagnosed with colorectal cancer. Analysis of the epigenome of this patient's colorectal cancer identified abnormal DNA methylation patterns of certain tumor suppressor genes in this patient's cancer cells. Which of the following best describes the effect of DNA methylation on gene expression in this context? Okay? Everyone's saying B. Good. So now I think it was Rida that asked the initial question about why I'm going through the tumor, um, why this is relevant to tumors and stuff, because this is actually how they like to ask you these questions, okay? The question is basically, what's the action of DNA methylation? It's a very simple question. You can solve it by just knowing this, by reading this sentence. But they'll confuse you with giving you this scenario by explaining to you what's a tumor suppressor gene, what's an oncogene, you know. So just to clear that up and allow you to kind of guys to focus on this question of what it is. And yeah, it's basically a very simple question. DNA methylation inhibits gene expression. That's all this question is. Okay? But they've kind of made it difficult. Why does a microRNA bind to the 3' UTR region in particular? Not sure. Not sure. Okay? So Reem, do you still want me to go through the microRNAs part or? Yes, okay. Sure. This is a microRNA. This is pre-miRNA. Forget about this pathway here, okay? This is pre-miRNA, the immature form of microRNA. It runs into an enzyme called dicer, which is an RNase enzyme. It shortens the mRNA. It kind of shaves off the unnecessary bits. And now you have mature microRNA. It complexes to proteins to form an RNA RIS complex. These proteins are specifically called Argonaute proteins. It's not important, okay? RNA RIS complex is what's formed. This RIS complex, RNA RIS complex, is complementary to a certain mRNA, a target mRNA. When it binds to it, it degrades it. Okay? That's it. So know dicer, know that microRNAs are a form of RNA interference, know this RIS complex, and know they degrade mRNAs. Okay? Okay, thank you very much. That's it.
Thank you so much, Dr.
Thank you.